All right, folks, it's time to get going, so we'll start. Questions about, we covered last time introduction to blood, what it's made of. We talked about the general adaptation response or the stress response, and then a little bit about growth and hormones. So anything on those topics before we move ahead? All right. Okay, well, I still don't have my videos won't play in PowerPoint, but I did figure out how to get them to play. So I'm just going to show you the one from last time. I know a lot of you saw it on your own computers, but it's still cool to see. Hold on, let me see where I made it big. So what you're seeing there is, that's a medium-sized vein. So you've got artery here and vein here. And you can see, if you look carefully, this video isn't very high quality, but you can see some of the little specks are actually red blood cells moving around. So we oftentimes think that blood flow in the body is slow. This is real time. So blood really flies through the vessels. And while it is true that the smaller the vessel, the slower the flow, even in these small vessels, the blood is still flowing very rapidly. So you can imagine in the aorta, which is the largest artery, how fast blood is flowing through there. I mean, it's like a bullet train compared to an interstate like this. So kind of cool pictures, I think. And I've got some other fun videos today we'll look at. All right, close that one. And go to here. Okay, so we talked last time about red blood cells, and essentially all a red blood cell is a cell membrane chock full of this molecule right here, which is called hemoglobin. So red blood cells are super specialized. Before they enter the circulation, at least in the normal case, they've lost their nucleus, and they've lost most of their other cellular organelles. So they don't have an endoplasmic reticulum, they don't have a nucleus, they don't have Golgi apparatus. They have a few mitochondria, but not very many. So they really are specialized carriers. And what they carry best is oxygen because of this molecule right here. So hemoglobin is a compound protein. So, a protein is a long chain of amino acids that's been twisted into a shape, and it's that shape that allows it to accomplish some fancy chemistry. Well, in the case of hemoglobin, it isn't one long chain of proteins or a long chain of amino acids. It's 4 connected together. So we have 2 alpha chains that sit diagonal across one another, so alpha 1 and alpha 2, and then we have 2 beta chains, beta 1 and beta 2. The alphas are the same, the betas are the same. So chain 1 and chain 2 are not different. So you've got 2 building blocks, alphas and betas, 4 of which come together to make this hemoglobin molecule. And the magic of hemoglobin happens at this disc-shaped molecule here called heme. Heme is not a protein, it's just attached to one. So each of these chains has this frisbee-shaped molecule attached to it in this kind of triangular crevice that evolved to accept it. Heme is a very strange molecule. I mean, this is its chemical formula. Remember, I said you don't have to know chemical formulas. But those of you who have had some chemistry, which should be all of you all, will notice that this is kind of a fancy molecule. We've got multiple rings all attached together. We've got nitrogen in here. Nitrogen has a tendency to make stronger bonds with things. So the short version is heme is a very, very strong molecule. So much so that our bodies cannot completely break it down. We can make heme, but we can't break heme, because once it comes together, all of these double bonds and ring structures make this a very solid molecule, hard to break metabolically. And the reason this heme molecule gets made is right in the center is an iron ion. So this is the same kind of iron that's found in, the cylinder block of your car, only this is the ionized form, so it can be dissolved in water. And the magic of this iron attached to this heme is this 2+ charge means that iron can hold on to an oxygen molecule. So, you all remember, and not an oxygen atom, but an oxygen molecule, right? So you've got your O, right? And because this iron has a positive charge, oxygen gets bent a little bit and kind of looks like this when it's attached to that spot. And the cool thing about this is this isn't a chemical reaction. Well, it's a chemical reaction, but it's not a covalent reaction. It's an ionic one, which means that this is reversible. So oxygen can be held by that ion, or it can be released by it, depending on the environment that this hemoglobin molecule exists in the body. And we're going to talk in the respiratory chapter about why that's important. But the short version is this iron in that heme molecule, it means oxygen can be gathered up where there's lots of it, like in the lungs, and then out of the tissues where there's not very much oxygen, it's released so that the cells can use that oxygen to make ATP by burning carbohydrates, lipids, and fats. All right, so I do want you to know alpha and beta chains, right? 4 subunits, so there's 2 alphas and 2 betas, heme and iron. Because there are a number of disorders of hemoglobin that are quite prevalent in the population. So if you've ever heard of thalassemia, for example, thalassemia is a defect in either the alpha chain or the beta chain, or both in the severest form. So it is important for you to know the pieces. But, the molecular structure of hemoglobin is way, way beyond our course. That's more of a biochemistry topic. All right. So it's all about the iron in the end. All right. So we'll pick up hemoglobin's story in the respiratory chapter when we talk about gas exchange, how oxygen and carbon dioxide are moved around the body. But we do need to talk a little bit about how red blood cells are produced and destroyed. So, hugely specialized, they've lost their nucleus. That means that red blood cells cannot repair themselves. Cells in our body that have a nucleus have the genetic code to replace any part of themselves with new proteins, new carbohydrates, new lipids, whatever. Because the red blood cell has lost that nucleus, it now has a limited lifespan. So red blood cells live about 120 days, so about 4 months. And, what happens when they're at the end of that 120 days? Well, one of a couple of things happens. All right, so we'll start here with the start airways. This is red blood cell production. Okay, in the bone marrow are these cells called proerythroblasts. And through a stem cell process, red blood cells are constantly produced. Okay, so these are not the stem cells you hear about on the news or on Facebook or the internet or whatever. These are stem cells specific to the blood system. And what a stem cell is this. You have a cell, right? So a cell, and it divides, okay? Because we know most cells can do that. So we get 2 daughter cells. In a stem cell system, one of these daughter cells becomes the original. So this is what we mean by a stem cell, is a system where through cellular division, something is made, right? So this cell gets made for some other purpose, but then the original cell is replaced. So that's all that term means, stem cell. Now the beauty of this is this can go on forever and ever and ever, right? Because as long as there's a cell to divide, one of those cells can go on and become something, another replaces the mother cell, and we have this perpetual cycle. So in this case, the proerythroblast, remember blast is to make, right? That's what that word means. So this, pro is before, erythrocyte means red cell, which is blood cell, right? So this is a cell that makes pre-red blood cells, right? Lego words? All right. So in this case, the proerythroblast, in the end, it creates a red blood cell out the other end. And that is kind of what we see here in this first box. So this cell divides, and then one of its daughters starts to differentiate. The nucleus gets smaller and smaller here, and then eventually it's ejected. So the nucleus goes and it's recycled by the bone marrow. The DNA and RNA that it contains are. And then what comes out eventually is this shiny new red blood cell with its classic flattened disc shape, right? So it does its job. It circulates around the body, 120 days. It travels some huge number of miles in that time, going through the vessels over and over again. It gathers up oxygen in the lungs. It dumps oxygen in the tissues. But eventually, all that travel, just like a good suitcase, eventually wear and tear starts to take hold, right? Some of our vessels are smaller than the red blood cells are, which means the red cells have to squeeze through these little vessels, and that eventually causes traumatic injury. And at some point, they've reached their natural life. As red blood cells get older, they get more and more rigid, less and less able to bend and get through those little nooks and crannies. So about 90% of red blood cells end up dying in just a couple of places, either in the liver, the spleen, or the bone marrow. Now, the reason they die there is those are some of the regions where the capillaries are the smallest. So if a red blood cell becomes less flexible as it gets older, at some point it's going to get stuck in the little nooks and crannies of the liver and spleen and bone marrow. So that's where it's going to die because that's where it gets stuck. Now, our bodies are excellent recyclers. So we're not going to let all that good stuff in the red cells go to waste. Instead, we're going to pull that red cell apart and shift its constituent pieces in different directions. So one of the most important components of red cells is the iron they contain. Iron is relatively scarce in biologic systems here on Earth, right? So it's not like there's boatloads of iron in anything that we eat. So in order to keep our iron needs low in terms of diet, we need to recycle as much iron as we can, which we can do because the nature of atoms is that they don't have wear and tear. Molecules break down. Atoms are forever, so to speak. So that same iron can keep getting recycled because it's unchanged. So the iron gets pulled off and it gets shipped back to the bone marrow where it's going to be incorporated then into new red blood cells. So there are some proteins that carry that. The principal one is transferrin. Ferrin means iron. Trans means transport. So this is a protein that transports iron from the spleen or the liver into the bone marrow for recycling. Well, what about the other pieces? Well, the heme does wear down. Okay, one of the reasons why red blood cells have a lifespan is that heme molecule, even though it's super tough, it does get worn out, so to speak. So we don't want to recycle it intact. Instead, we want to build a new heme and get rid of the old heme. But we can't break that molecule because we just don't have the metabolic machinery to bust all of these double bonds and rings. So instead, we kind of warp it. We take heme and we bend it and squish it a little bit, and we get first biliverdin and then bilirubin. And the reason we do this partial metabolism is to make it soluble in something. In this case, lipid. So bilirubin is lipid soluble. It's a tiny bit water soluble, but not a significant amount. So the heme goes to bilirubin. The bilirubin goes to the liver, and then because bilirubin isn't very water soluble, the liver actually excretes it into the small intestine through the bile ducts, much more on that in another chapter, and it goes into the small intestine, and then it's eliminated along with the feces. So bilirubin is removed from the body via the digestive tract. And we're going to see there's a couple of things where the digestive tract is the source of elimination. Typically things leave the body through the kidneys. But things that aren't water soluble, the kidney can't deal with. So it's up to the liver to get rid of it by putting it into the digestive tract instead. Now, bilirubin has a color. It's shown here in green. It's because it is green. It's bright green, actually. And some of its oxidized forms are kind of brownish green. But one of the reasons for the color of bile, which is very green, is the bilirubin that it contains, because it's actually a pigment. People used to use ox gall, so the bile from oxen, they used it as a component of paint to make bright green paint because it's bright green stuff. So that's the path of heme, goes to bilirubin, and then out. There are a ton of disorders associated with bilirubin that you're going to learn about later. When your bilirubin level is high, you turn kind of a yellowish-green color. It's pretty prominent if you get to see that in a patient. Or if one of your friends has a patient with jaundice, bring your other friends to go look, because it's something you can notice from the door about people, particularly people with really light skin, because they turn like green, really profoundly green. So that's any elevation of bilirubin causes that jaundice effect. There are a ton of reasons why you can get that. You know, if your liver is failing, this part doesn't work, so the bilirubin backs up. If your red blood cells are dying at an increased rate, like you see in sickle cell disease, then you have more bilirubin, and that causes jaundice, so there's lots of different varieties and variations there. Okay. Well, we have some little arrows here, too. About 10% of red blood cells don't make it in this normal pathway. They get broken somewhere else along the way. But in the end, their pieces still end up being transported to the liver, where the iron is pulled off and the heme is metabolized. So not all of them make it all the way back to home base, so to speak. Now, the kidney does secrete a little bit of hemoglobin. In the normal case, you should have almost none. But let's say you've had some traumatic injury. You know, you were in a car accident, or even better, a crush injury, like something heavy fell on you. That causes a bunch of red blood cells to break, and you can see brown or reddish urine for days after that. And that's the hemoglobin that's circulating in the bloodstream free is being removed from the kidney, or by the kidney. And then a little bit of the bilirubin is sent to the kidney too as urobilins. When you do your urinalysis in lab this semester, that's one of the things you're going to look for is the urobilinogens, they're called. So red blood cell life cycle. The important piece is the recycling, the breakdown of heme, because like I said, there's a bunch of disorders that affect this spot right here. And how our iron, most of the iron that we use every day, we didn't take in by mouth. It's recycled from our red cells. Yes?
So is the ultimate function of bilirubin just to wash away the broken
to get rid of the heme. It is the old heme. It's like heme that's been hit with a sledgehammer. That's all bilirubin really is.
So it's essentially just waste.
It's a waste product.
Okay.
Yeah.
Okay.
Yeah. And we make it all the time because our red cells are dying all the time.
Okay.
Yeah. And we make it in bigger amounts than you might think. I mean, one of the reasons that feces is the color it is because of all the bilirubin that's present. You know, you wouldn't think, well, only 1/120th of all red cells die in a given day, but that's still like 500 million red cells, so there's a lot of bilirubin that gets made. Okay. Good. Okay, we can't talk about, yes?
So is some of the heme still recycled?
Heme is not usually recycled. Some of it is, but mostly it's broken down to bilirubin and made fresh again. Yeah. In some ways you'd think, well, why don't we just recycle it? And it's because old heme doesn't hold the iron as well as new heme does. So it has to be remade. Yeah. All right, so blood type. You've all heard of this. Who's never ever heard of blood typing ever in their life before? Okay, good, because I'm going to make this sort of quick then. All right, so the key things to remember, your blood type is what your blood cells have on their surface. So it names the antigen. An antigen, for now, is just a shape. Okay, remember, a lot of biological chemistry is about shape. In chemistry class, you watch atoms move around. Well, in biology, the chemistry of biology is really about shapes moving and changing. So an antigen is just a shape that's found on all of our cells, but in particular, red cells don't have very many shapes on their surface. They don't have very many antigens. So the ones they do have matter a lot. Okay? So if type A blood has the A antigen, type B blood has the B antigen, type AB does not have the AB antigen. It has A and B, right? And then type O does not have the type O antigen. It doesn't have, O stands for nothing, for null. It doesn't have A or B. So you can think of it as just like an O is smooth, a type O blood cell is smooth too. It doesn't have shapes on its surface, at least not of these types. Well, so that seems easy enough. So there's 4 blood types. They declare what's on the surface. The complicated bit comes in the fact that our bodies make antibodies to any shape that our own cells don't have. So what that means is if you have type A blood, you will have antibodies to type B blood. So the opposite. You don't have that shape, so you have antibodies to it. Type B has type A antigens. Type AB, you see empty here. It's because what antigen or what antibodies could you make when both antigens are present? So there's no antigens here. And then in type O, they have both antigens to A and antigens to B because they have neither one. So this table right here declares which type of blood can be given to which type of patient. Okay, so for example, type AB blood, they don't have any antigens. So you can give them A, B, or O because they don't have anything to react with these antigen shapes. So we call that the universal recipient. If you have type AB blood, you can get blood from anybody. Okay? Type O blood is the opposite. They have antigens to both A and B, so they can't get type A because that would react with this. They can't get type B because that would react with this. And they can't get type AB because it would react with both. Now, what do I mean by react? It's not subtle. If you give type B blood to a type O patient, you're probably going to make them sicker than they were when you gave them the blood in the first place. Because in a bag of blood, there's hundreds of millions of red cells. And you just put in a massive, it's like being massively infected all at once. So they have immune system fires up, cardiovascular collapse. People can die from this, which is one of the reasons everybody learns the typing system. All right. Now, there is another version of this, which you're going to learn about in lab, and that's the Rh factor. So in addition to antigen A and B, we also track the Rh antigen. If you're Rh positive, it means you have the Rh antigen on your blood cells. If you're Rh negative, it means you don't. But the same rules still apply. It's just one more factor that we use in everyday blood matching. All right, so hopefully that's all things you've heard about. You're going to type your own blood in lab. So you'll get to be up close and personal with this in, probably next week. Or have you done it already? No? Yes?
What's the difference between O positive and O negative?
O positive has the Rh factor, and O negative doesn't. So the true universal donor is O negative, because they have nice smooth red blood cells, so there's nothing to attach. Yes.
So what does it mean when you have the Rh? What does it mean when you have the Rh factor?
It doesn't mean anything. It just means you have that, which means you can't or you shouldn't give blood to somebody who doesn't have that Rh factor. That's all it means.
So if you gave like type A positive to a type A negative person, would it react like that?
Potentially, yes. Rh is a little softer. It isn't as vigorous as this, but particularly if they've had a blood transfusion in the past, they'll have a nasty reaction. Yes.
Why are there different blood types? Like,
Just genetic variation. That's all.
Wouldn't it make sense just to have one?
Well, yeah, but it didn't evolve like that.
Yeah.
And there's cultural or there's genetic heritage difference here. So like people whose genes are from one part of the world have more AB, for example. Other parts of the world have more O. So it's just classic, just like hair color's different and eye color's different and all that.
Do animals have blood types
Yep.
as well?
They do. This system is present, I think this is in all the mammals. Not the Rh, but the AB part is. So yes, and nowadays in veterinary medicine, we actually give blood transfusion to animals. It didn't used to be like that, but now we do. All right.
Is it from human to animal? Or
No, it's animal.
Okay.
People played around with that. Can we take blood from an animal and save a human with it? And There's so many differences between the red cells that ends up causing a massive immune response.
And vice versa.
And vice versa, yep. Species are very specific, particularly at the biochemical level. Yeah. All right, so blood type in a nutshell. So what does happen if you give blood to somebody with antigens? You get this, right? You can imagine red cells all stuck together, probably not going to circulate very well, right? And then antibodies trigger an immune response. So not long after this, we get this. We get red blood cell pieces everywhere. And now you've got a patient whose blood is basically breaking down. So you have an emergency. All right. And this is what you're going to see in lab, so let's get that. Okay, a question. Formed elements. So that's the part that isn't plasma. Formed elements make up about what percentage of the blood by volume? Everybody able to get on? Yes. Like it? All right, so the standout is C. Here, let's go back a couple of slides. So the best answer there is, it's about 60-40, right? So 45 was one of your answers, so 45 formed elements. So the plasma is always the larger amount, okay? So I always think of it as 60-40, but technically you could have half and half, although that would be abnormal. Usually there's much more plasma than there is formed elements. So in our question there, the best answer would be that 45 or D. All right, so look at that again because 67% of you put C on that. All right, which of the following is not a component of plasma? Ooh, it's all over the map. I got answers in every column. All right, so plasma is the part of the blood that isn't inside a cell membrane. Okay, so you've got cell membranes on red blood cells, cell membranes on white blood cells, and cell membranes on platelets. So the one of these that doesn't belong as plasma is B, platelets. It's part of the formed elements. When you put it in a centrifuge and you spin it down, the platelets pack down there with the red cells and the white cells. So it's not part of the liquid part, plasma. All right. Okay, so red blood cells, that's 99% of what's circulating in the formed elements. But the white blood cells play a major role in the immune system. Okay, so one thing to know for sure about white blood cells is this first point. Even though we talk about white blood cells in the blood, we measure them in the blood, they have blood in their name, the white cells, they don't live in the blood. So any white cells you see in the blood are just on their way somewhere. It would be like measuring traffic on Dodge and saying that people lived where they were measured in their cars, right? Of course that's not true. You're just going somewhere. Same thing for white cells in the bloodstream. All the white cells are immune system cells, And their job is to take care of all the cells of the body, the whole body. So just because we find them in the blood doesn't mean they live there. They're just on their way somewhere else. Okay, so that's one aspect of white blood cells. Another is, kind of goes with the first one, white blood cells can all get out of the blood vessels too. Red cells are pretty much trapped inside the vessel system. Unless there's a break in a vessel wall, red cells can only be in the heart or the blood vessels. They can't get out. But white blood cells can, and they do so through a process called diapedesis, which means 2 foot. Dia is 2, pedis, like pedestrian, is foot. So we have diapedesis. All right, so here's our white cell, right? And it's cruising along. Out here is some damage, right? So we've got, these are cells all in here. So you sprained your ankle, let's say. All right, so the white blood cells, they come along, they attach to the side of the blood vessel, and then there's these little holes in the capillaries, and what happens is they squeeze themselves through like that because they're squishy. You know, it's like the blob. If a giant amoeba tried to come in through that door, it could because it can squish itself in and out. And at some point, it looks like it has 2 feet, right, because it's going through this narrow space. So we call that diapedesis. And the process of this white cell leaving the bloodstream, because then it goes back to looking like that, we call that emigration, which is right here. So this is emigration. And it sort of fits together. If white cells use the bloodstream to get to places, it means they have to be able to get into the bloodstream and out of it. Otherwise, how could they use it for transport? And they can't through this process of diapedesis. Okay. Now, how does the white cell know that this is where it should get out, that this is where it should turn off into the neighborhood, so to speak? Well, when there's damage, sorry, that's bad handwriting, damage, right? We get chemicals that are released. Those chemicals attract white blood cells. So we call that chemotaxis. That word's on here somewhere, right here, right? Taxis, like a taxi gets you from place to place. Chemotaxis is motion because of a chemical. It's basically following a chemical trail, you know, just like a bloodhound can do. You know, they're following a molecular trail to get the bad guy. Well, in this case, white cells sense these chemicals and are attracted to them. So they leave the bloodstream and then they move out to where they're going to do their job. And then 3, neutrophils, eosinophils, and monocytes, we're going to talk about all 5 types in a minute, are also capable of phagocytosis. That means once they've left the bloodstream, they've been attracted to this area of damage, some of those 3 cells can swallow other cells, particularly bacteria or damaged cells. One of the ways the body destroys stuff, as gross as it is to think about, is it eats it and digests it, right? So these cells swallow these bacteria and digest them, and now they're dead bacteria, so they can't do any more harm, right? And I got some movies to show you some of these motions. All right, so these are common characteristics, but we divide the white cells into 5 groups. All right. So we have our white cells. And what I expect you to know about the white cells is their names, what they do, and what I call their claim to fame. In other words, the thing that makes each white cell so unique. All right, so we have neutrophils, eosinophils, and basophils. And this group are called PMNs. So I've written out what that means. Polymorphonuclear granulocytes. That's PMNs, also lovingly referred to as polys. Now, what on earth does that alphabet soup mean? Okay, take it apart. Poly is many, right, like polygon. Morpho is shape, you know, like to morph from one shape into another, or morphology is a word we use in science, the shape of the thing. Nuclear is nucleus, right? And granulo is granules, as in grains of sand, like grains of something. Cytes is cells. So what that tells you is these are cells that have granules in their cytoplasm, and their nuclei have many shapes, right? So polymorphonuclear granulocytes. An example is here, here, and here. Do you see how their nuclei have these funny shapes? Right? That's how they get their name. And if you look, do you see the little dots in here? Little dots in here, little dots in that one. Those are the granules. So they're named according to their appearance under the microscope. All right, and then the other two, the agranulocytes. Granulocyte means cell with grains in it. We put an A in front, it means no grains. So these are cells with no grains in them. And there's two of those. There's monocytes, lovingly referred to as monos, and there's lymphocytes. So those are the 5 categories. Okay. And we're going to go into some detail about those in a minute, but first, some cool movies. All right. So chemotaxis is the ability of these cells to move and follow a chemical trail. All right? Their movement is most like an amoeba. Everybody remember looking at amoebas in high school or micro? All right. Here we go. Okay, so this is the cell in question, right? We're gonna watch it hunt. Okay, so here's the bacteria, right? And it's cruising around. It's following a chemical trail that this thing is leaving behind it. So just like a terrier running after a rabbit, it's the same kind of thing. And this cell will pursue this bacteria until, like it's doing right now, until it's able to swallow it. And then eventually you'll see that little black mark disappear as it's digested. And then it's off in search of its next target, right? All right, so that's one. That's chemotaxis. And then the other one.
Is that real time? Like they didn't speed up?
No, that's real time. Yeah. They're quick. But remember, they're tiny too, so they're not moving very far. So this is that's a macrophage, one of the largest phagocytic cells in the body. And guess where its target is? So here it comes, swallow the thing whole. Right now it's in a little digestive vacuole. And then eventually, boop, there it goes. Where'd it go? It's gone. It got digested. So this cell will then excrete the parts of that it couldn't use. What it can use, it'll use as nutrients. So our immune system actually feeds on the infections that it fights off. Most of their nutrients are still gotten from somewhere else. But so you can see it's still chemotaxing. And, this kind of movement means it can move in any direction. So like for us, because we can't change our shape like these guys can, if we want to walk, to the side, we have to turn and then walk. They can just move that way or even up and down. So it's a very effective means of locomotion. All right. So that's these characteristics, right? The shared properties. All right, so let's get into our short list. Okay, so the polymorphonuclear granulocytes, the PMNs they're called, or polys they're called. The first up is the neutrophil. The neutrophil is the most prevalent white blood cell in the body. There's more neutrophils than any other kind of cell. And neutrophils can dramatically increase their number very rapidly. And when you think neutrophil, think phagocyte. In other words, it's an eater of things. And think bacteria. Okay, when you have a bacterial infection, neutrophils are the principal attacker, at least at the beginning of that infection. So, you stepped on the classic rusty nail, right? within 2 hours of that infection or that bacteria introduction into your body, your neutrophil count will go way up as neutrophils come flooding out of the bone marrow to attack whatever this infection is. So it's one way in laboratory medicine, we can measure the numbers of all of these different cell types individually. And so if we get a lab report and the neutrophil count is super high, we're going to start looking for a bacterial infection somewhere, right? Because neutrophils go with bacteria. Now, in terms of appearance, which you should be able to know by sight or by description, right? The neutrophil it's clear for the most part. Its nucleus stains a deep purple, and it usually has 3 or 4 lobes to it. So this is all nucleus here. You see why they call them polymorphonuclear, because their nuclei really are funny shaped. Usually nuclei are round. Yeah?
So when you say it says that there's more neutrophils on like a blood test.
Call that a CBC, a complete blood count.
Okay, so does that mean there are more, like more were made, or they're more active?
No, there are more. The physical number goes up.
So
because the bone marrow, the bone marrow keeps a reserve of neutrophils that are ready to go.
Oh, okay.
So yes, you actually have more neutrophils in your body at that time.
Okay.
Yeah, because they're coming out of the bone marrow to fight the infection. Yes.
Which is a better determinant for bacterial infection? Would it be bands, which are precursors for neutrophils, or neutrophil itself?
Bands.
Mm-hmm.
Bands are those cells that are coming out of the bone marrow real fast. So yes, unless it's all bands, and then you're going to be looking at cancer, leukemia. But yes, bands are even more a better indicator than neutrophils are. Usually the two go together. So yeah, good question. We're not going to talk about bands. All right. So clear, speckled cytoplasm, deep purple nucleus with multiple lobes. Think phagocytic, and in particular bacteria, which I'm not sure if they even mentioned that in this, but that's a good thing for you to know. Okay, eosinophils. Okay, why do they get their name? Phil is like, or so a neutrophil has a nucleus that likes staining, right? So they call it a neutrophil. Eosinophils. Eosin is a kind of bright orange stain that we use to look at things. So in most pictures, your eosinophils are going to be a strikingly bright orange or pink even, okay? Their nucleus usually has kind of a C shape right here. And the claim to fame here is eosinophils are associated with allergies and parasites, okay? Eosinophils evolved to fight off invaders that are larger than cells, is the idea. So we don't see this in this country very much anymore, but our ancient ancestors had to fight off parasitic worms, right, in their digestive tract, in their skin, in their mucous membranes. The eosinophils played the largest role in combating those large, even macroscopic creatures. So eosinophils go with parasites, and the allergic response is often, eosinophils play a role in that. So like if you're a person who has hay fever and asthma and eczema, your eosinophil count all the time is elevated. Now, why? One theory is as we defeated the parasites, the macroscopic parasites like worms, it left a part of our immune system with very little to do, right? And one theory is that the eosinophils now overreact to things because they aren't busy doing something else. So it's just one theory. Have you ever heard the spoonful of dirt theory? That in some ways in modern America we'd be better to have a spoonful of dirt every day because it would keep our immune system the way it should be. But I don't think anybody's going to do that. All right. So that's eosinophils, parasites, and allergies. Basophils, they're dark purple. So dark purple that you really can't see the nucleus on the inside. It's in there, and it has a shape similar to this, but you usually can't see it. Instead, it's just the granules are so dark you can't see anything else. And basophils, I want you to associate with this word right here, histamine. Okay? Basophils produce histamine. Histamine is a primary chemical regulator of inflammation. Inflammation is the body's general response to an attack or an injury. It's easier to just describe it. Inflammation is redness, swelling, warmth, and pain, right? So we've all twisted our ankle once or twice, right? And we get red, it gets red, it gets swollen, it gets warm to the touch, and it hurts. That's inflammation. That's the body's answer to any kind of injury, attack, infection, whatever. And when we get to the immune system, we'll talk about inflammation and why it exists. But just let me ask, who's had immunology? You guys are going to be bored with some of this immune system stuff, but it'll be a good refresher anyway. All right. Okay, so these three are the polymorphonuclear granulocytes. They have their appearance, and they have their claim to fame. All right, what about the other two? Monocytes are sort of boring-looking cells. They look like the typical cell that you see in cell biology textbooks, right? So they have a medium-sized nucleus, some cytoplasm. There really aren't granules in it. Their cytoplasm is pretty clear. And the thing I want you to know about monocytes is they are baby macrophages. So macrophages are cells that live in the tissues, and they get huge. That big cell we saw, that was a macrophage. And they're phagocytic cells. So they gobble up things that don't belong, or they gobble up debris. Monocytes are baby ones of those. So they're going to leave the bloodstream. They're going to set up a home in some tissue. They're going to get giant, and they're going to eat anything that doesn't belong. It's sort of what a macrophage does. And then the lymphocytes are a little more than a giant nucleus with a little tiny bit of cytoplasm around it. Now, the lymphocytes, the claim to fame for today anyway, is they are the boring-looking cells that do the most complicated stuff. So all the fancy stuff our immune system does is all lymphocytes in action. So we're going to cover that when we get to the lymphocyte chapter in the next unit. And then the last of the formed elements are these guys. So the platelets, essentially under the microscope, they look like little pieces of broken glass. You know, they have no particular shape. They have sharp edges and round edges, just like if you dropped a plate of glass on the floor. And platelets I want you to associate with coagulation. So they play a significant role in blood clotting, which we'll talk about hopefully next time. Now, where do they come from? Platelets are shards or pieces of giant cells called megakaryocytes that live in the bone marrow. So instead of our bodies having coagulation cells, because we have to have a bunch of them and there's already red cells and it would be crowded, instead we just have these little pieces of cells that do that job instead. So like platelets are very small, and most microscope pictures, the scale is so small that you barely see them. You know, like in this picture, there aren't even any platelets, but it would look like a speck on this scale. So they're very, very small. But they do play a major role in blood clotting. To give you an example, when patients have a platelet count of 0 or very low, they spontaneously bleed because the platelets are so necessary to keep all of the little tiny tears in blood vessels from hemorrhaging. So they're critical to have, even though they're these little tiny things. All right. And here we'll do this last piece. You can pretty much cross this one off from a learning list, right? What this is showing you is the family tree of, here's our formed elements. We already talked about this one. This one you are responsible for, okay, the red blood cell one, because it comes up. So proerythroblast, and then to reticulocyte, and then erythrocyte. This is the platelet pathway. Here's that megakaryocyte, great big giant cell. And from this end, it sheds little pieces of itself, and those become platelets. But these, while this will be important for you to know when you get to your hematology section in your PA studies, for today it's a little much, so we're not going to look at those. This is just where do these 5 types of red blood cells come from. And essentially they all have a common ancestor. The lymphoid system has one, and then the myeloid system has another, but like I say, no need to memorize this, as I say right here. Okay. Just as a parting note, today we have chemicals that we can inject into people that promote the development of one type of cell or another. Okay, probably the most common one is erythropoietin, EPO. If you give somebody erythropoietin, their red blood cell production goes up. and I'll talk about why that's true in the renal system. But we also have similar chemicals that will promote white blood cell development too. So let's say you've got a patient with cancer and they're on chemotherapy. Chemotherapy wipes out white blood cells first, right, because they're rapidly dividing cells. So one of the tricks we have is as their white blood cell count starts to tank, we can give them stimulating factors that help to promote the bone marrow to produce more white blood cells. But, all right, that's it. We're out of time. We'll pick up on coagulation next time. Questions about anything? I know we're out of time.